Hello everyone. I am Dr. Deepak, currently working as an assistant consultant in Department of Anaesthesia in Saudi Arabia. I have done my MD post graduation from Safdarjung Hospital, New Delhi, India. And just like you guys, I am a USMLE aspirant too, and I am preparing for USMLE Step 1 currently. So I have created this YouTube channel for the discussion and preparation for USMLE step. And today we will start our topic from general microbiology. We will try to cover the topics from first aid, Kaplan notes, UWorld, Emboss, and USMLE RX, which are supposed to be the best resources for the preparation of USMLE step 1. Okay, guys, so we will start with general microbiology and we will move on to the next topic in a stepwise manner. Our main learning objective in the general microbiology will be to understand basic things like bacterial cell wall structure and what is the function of the bacterial cell wall. Then we will gradually go into the details of the gram positive and gram negative cell wall structures, their differences, right? Bacterial cell wall, right? So bacterial cell wall, it is a tough and rigid structure, right? It is a tough and rigid structure, and it surrounds the bacterium. It surrounds the bacteria. It surrounds the bacteria, and it is around 10 to 25 nanometer thick, right? It is 10 to 25 nanometer thick, and it constitutes around 20 to 25% of, I will say, dry weight, right? Dry weight of bacterial cell, dry weight of the bacterial cell. So this is basic bacterial cell wall introduction. Bacterial cell wall is a tough and rigid structure which surrounds the bacteria. It is around 10 to 25 nanometer thick, and it is 20 to 25% of the dry weight of a bacterial cell. Now we will understand the basic thing that what is the function, what is the function of cell wall. So we will understand in terms of the headlines. First is bacterial cell wall has a very important role to protect the bacteria against the osmotic lysis. Osmotic lysis protection, right? Osmotic lysis protection is nothing but it is a protection against the process of osmosis. Osmosis is the movement of the fluid across a semi-permeable membrane against a concentration gradient. And when a bacterial cell is in some adverse condition, like in bacterial cell is in hypertonic or hypotonic solution, or hypotonic surroundings, it is the bacterial cell wall which provides protection against the osmotic damage. Okay, second thing is the rigidity. It provides rigidity. Rigidity, how come? Rigidity is because of a very important part of the bacterial cell wall. We call it peptidoglycan. Peptidoglycan is a structure. Peptidoglycan is a layer, you can say, in a bacterial cell wall that provides rigidity. And bacterial cell wall is responsible for the shape of the cell, right? Bacterial cell wall has important role in the cell division. We will learn about the bacterial cell wall cell division, which is very basic kind of reproduction method. By the method of binary fission, we will learn about it a little bit. We will not go in detail, and that is where the bacterial cell wall find its role. The another important role of bacterial cell wall is to protect, you know, protect against toxic substances. Toxic substances. Any toxic substance, bacterial cell wall is protected by its virtue of cell wall. Bacterial cell wall is a site for action of, I hope you understand, action of antimicrobial agents, or what we normally called antibiotics. So it is a cell wall which is the primary target for many classes of antibiotics, like the penicillin group of drugs, say for example. So bacterial cell wall is also the site of action of antimicrobial agents. I will show you one pictorial depiction from the Kaplan, which shows a very brief idea about the various classes of the antimicrobial agents which act at the different sites on the bacterial structure, and few class of antimicrobial agents target bacterial cell wall. Okay, next function of bacterial cell wall, you can say, is the virulence, very important, virulence factor. What is virulence factor? Virulence factor, it is the reason which contributes to the pathogenicity. It contributes to the disease causing factor. You can say virulence factor is a disease causing factor in the bacteria, and this disease causing factor is actually because of the presence of certain structures on the cell wall of the bacteria. Like example of virulence factor is, can you say enumerate few examples? Yes, it is endotoxins. One of them is LPS. LPS stands for lipopolysaccharide, and endotoxins. These are few examples of virulence factors. We will go in detail because this is very important topic from the step 1 point of view, USMLE step 1. Okay, and the next role or function of the cell wall is providing immunity, right? how come immunity how come immunity is provided by bacterial cell wall to the bacteria itself? Immunity against, I will say, immunity against the antibody which is generated against, right, generated against the cell wall antigen, right? So it is the immunity provider, so what I'm trying to tell you is that bacterial cell has several roles. Osmotic lysis protection. Bacterial cell wall provides rigidity because there is a specific layer in the bacterial cell wall called peptidoglycan. We will discuss it. Peptidoglycan layer is responsible for providing rigidity to the bacterial cell. Particular shape of bacteria is because of the cell wall. Cell division, that is binary fission, is facilitated by bacterial cell wall. Bacterial cell wall provides protection against the toxic substances. Bacterial cell wall is the site of action for various antimicrobial agents like antibiotics, for example penicillins. And virulence factor, bacterial cell wall, is responsible for the you can say that bacterial cell wall has been attributed for the virulence or pathogenicity of the bacteria. Like example is endotoxin and lipopolysaccharide. Immunity, as in our body there are defense mechanism in the form of lymphocytes, macrophages, which provide immunity against the disease. Similarly, in the bacterial cell wall plays a very important role in providing immunity against the antibody generated against the cell wall specific antigen. Any example of cell wall specific antigen? Like lipopolysaccharide itself. Yes, it is LPS itself. Okay, so we will see that. Now we will go into another slide and we will see one by one the gram positive and gram positive cell wall structure. Then we will see gram negative cell wall structure, and then we will discuss the difference between the two. So if I am asked to put it in a very simple way, I will say gram positive cell wall is much simpler Then gram negative cell wall, right? Gram negative cell wall. It has three basic structures. We can divide it into two basic structures. We can divide it into like two components. One is the very important, I will say, peptidoglycan. Peptidoglycan. Okay. Another thing is we called teichoic acid. Teichoic acid, right? One thing I forgot to mention is that in the gram-positive bacteria, cell wall is around how much thick? 50 to 100, right? 50 to 100 layers thick. 50 to 100 layers thick, or you can say it is around 60 to 80 nanometer thick. Okay, so gram positive cell wall is much simpler than gram negative cell wall. It is around 60 to 80 nanometer thick. Okay, and the basic components are peptidoglycan and the teichoic acid. Peptidoglycan, what is this peptidoglycan? Peptidoglycan is a mucopeptide chain. It is a mucopeptide chain. I will show you the diagram of peptidoglycan for a better understanding. Mucopeptide chain, which is composed of alternating units of uh, N-acetyl-muramic acid and N-acetyl-glucosamine, right? Mucopeptide chain composed of alternating units of N-acetyl-muramic acid and N-acetyl-glucosamine. They are cross-linked to each other. They are cross-linked to each other, right? Each other. They are cross-linked to each other via what? Via tetrapeptide side chain. Via tetrapeptide side chain and pentaglycine bridges and pentaglycine bridges. We call it bridges. Don't worry, I will show you the diagram for a good understanding. Again, I am repeating, very important structure is peptidoglycan. In a gram-positive bacteria, it is also present in gram-negative bacteria, and we will see the difference in terms of peptidoglycan layer in the cell wall in among the positive and negative. Here I am discussing about peptidoglycan layer in gram-positive bacteria. In gram-positive cell wall, peptidoglycan is a mucopeptide chain which consists of the alternating units of N-acetyl-muramic acid and N-acetyl-glucosamine, and they are cross-linked. You know, these two units are cross-linked to each other via tetrapeptide, right? One is the tetrapeptide, another is pentaglycine, right? So tetrapeptide side chain and the pentaglycine pentaglycine bridges. um, Tetrapeptide side chain ascend from the NAM molecule. This tetrapeptide peptide side chain, it ascends from NAM chain. I hope you understand. I will show you a diagram so that you understand. Here it is. On my left side, we have got the structure of gram-positive cell wall, and on the right side we have peptidoglycan layer of gram-negative cell wall. So see, first gram-positive cell wall peptidoglycan layer on the left side. Okay, you see here G stands for the N-acetyl-glucosamine unit. M stands for N-acetylmuramic acid unit. As I said, in peptidoglycan layer, NAG and NAM, abbreviation for these two are NAG and NAM. These NAG and NAM are cross-linked to each other via the tetrapeptide side chain. Which one is tetrapeptide side chain? These four. Okay, these four is tetrapeptide side chain, as shown here in this diagram by small circles in column. These four rounded circles. Okay, and these are in a specific sequence. These are actually a set of amino acids which are in particular sequence. The topmost, which is attached to the NAM, is L-alanine. Second one is D-glutamine. Third one is L-lysine, and the last one is D-alanine. D-alanine. Okay, so this is the tetrapeptide, because it consists of 4 amino acids, so we call it tetrapeptide. Tetrapeptide helps in cross linking of NAG and NAM. How come? Because see, NAM is attached to the L-alanine of this tetrapeptide, and the other end of this tetrapeptide, that is D-alanine, is attached via this purple color circle, which represents the pentaglycine bridge to another successive or consecutive NAG molecule here. See, so this is the tetrapeptide side chain, because it is on the side of NAM, and there is a cross bridge which is made up of pentaglycine, pentaglycine bridge. Okay, so this is, and on the other hand, if you see the peptidoglycan layer of the gram negative cell wall, you can easily observe some basic differences. What are these basic differences? See, NAG and NAM are already present in gram negative also. In terms of presence of the units of peptidoglycan layer, there is no difference. Okay, we can also see the tetrapeptide. Okay, tetrapeptide. But if you notice, the tetrapeptide is not the same as the tetrapeptide in gram positive. How come? Because in tetrapeptide of gram negative bacteria, at the third position, amino acid is not L-lysine. It is, instead of L-lysine, it is mesodiaminopimelic acid. That's the difference. Mesodiaminopimelic acid. Okay, and one more difference we can easily observe here. There is no cross bridges. Pentaglycine cross bridge is absent here. We can see purple color pentaglycine cross bridges in gram positive bacteria. No, it's not here. The cross bridge is lacking. It's missing here in the gram negative bacteria. Okay, so these are the basic differences in the peptidoglycan layer. Another thing is teichoic acid. We were talking about another component, which is teichoic acid. First of all, I would like to say this is absent in gram negative. There is no teichoic acid in gram negative bacteria. And what is teichoic acid? Simple. It is polymer of the glycerol, or better called ribitol. Okay, that is not of much our concern. We just have to remember there is a structure called teichoic acid in the gram positive cell wall, which is absent in gram negative cell wall. That's it. Yeah, it may be important in maintaining the structure of the cell wall again. Okay, teichoic acid may be of two types, you can say. One is lipoteichoic acid, another is cell wall teichoic acid, cell wall teichoic acid. Okay, lipoteichoic acid and cell wall teichoic acid, right? So this is the gram positive cell wall structure. Now we will come to the gram negative cell wall structure. In the gram negative cell wall, gram negative cell wall, as I had mentioned in the gram positive cell wall, that is much simpler than gram negative cell wall. Yes, I am correct. Why? We will see here. Gram negative cell wall is much thinner, right? The moment I say it is much thinner, it sounds like it is much simpler also, but it's not like that. It is thinner, but it is more complex, and we will see in diagram. It is more complex, thinner but more complex. The different parts of the gram negative cell wall are, okay, I will show you one by one. One is the, of course, peptidoglycan. Another structure is OM, stands for outer membrane, right? Then one more structure is LPS, lipopolysaccharide. Okay, lipopolysaccharide, and last one is periplasmic space, you can say periplasmic space. So peptidoglycan is common in both positive and negative, but still we saw the differences. Peptidoglycan here, it is only 1 to 2 layer, uh, uh, around hardly 2 nanometer thick. It is very thin as compared to gram positive bacteria, where we saw it was around, sorry, in the gram positive bacteria it was 16 to 18 nanometer thick, right? The whole cell wall was 16 to 18 nanometer thick, and peptidoglycan was also quite thick in gram positive. But in the gram negative bacteria, it is only 2 nanometer thick. And at another difference we had already seen in the tetrapeptide, in the tetrapeptide side chain, tetrapeptide side chain, third amino acid was not L-lysine, it was something else. It was meso diamino pymalic acid, right? It is again not very important from USMLE point of view, but just to remember the basic difference in the structure of the peptidoglycan, I will say tetrapeptide side chain is chemically different in the gram positive and gram negative bacteria. Okay, so, and this is meso diaminopymalic acid, which is attached to the NAM unit. NAM is, sorry, NAM is the uh, N-acetyl uh, muramic acid. N-acetyl muramic acid. Let me um, erase it for you. Okay. Okay. So N-acetyl muramic acid unit. Now, tetrapeptide side chain directly linked to each other. One more difference. Tetrapeptide side chain is tetrapeptide side chain is directly linked to each other, right? Directly linked to each other means what? Means no pentaglycine side chain. There is no pentaglycine bridge, you can say. No pentaglycine bridge. Okay, that's it. Next is outer membrane. What is this outer membrane? Outer membrane is a phospholipid layer. It is phospholipid. Okay, it is phospholipid. It is attached to peptidoglycan by, it is attached to peptidoglycan, right? But we did not see anything like that in the case of gram positive cell wall, outer membrane. And it is a protective barrier, and outer membrane uh, consists of some specialized proteins, okay, called outer membrane proteins. Okay, outer membrane proteins, we can call it porine proteins. We can also call it porine proteins, right? Porine proteins. So we have porine proteins or outer membrane proteins. Very important structure in the gram negative bacteria is LPS. LPS, what is it? It is unique to the, I will say, it is unique to the gram negative bacteria. We will discuss here. LPS. LPS stands for lipopolysaccharide. It is unique to unique to gram negative bacteria, gram negative. Okay, it has three parts actually, right? It has three parts. The most important part of the LPS is lipid A. LPA most important part is lipid A, or also called endotoxin, right? Most important part is lipid A or endotoxin. it it is important. Why it is important? Because it shows endotoxic activities, and it is buried in, it is in, you can say it is in outer membrane. It is in the buried in the outer membrane. And um, it is responsible for the various things. I hope you know, or you will understand it later when we will go in detail about the individual groups of bacteria: gram positive cocci, gram negative cocci, gram positive, gram negative bacilli. We will learn gradually that this lipid A or endotoxin is responsible for pyrogenicity. Okay, pyrogenicity. It is responsible for the lethal effect. For the time being, you can remember these terms: pyrogenicity, lethal effect. It is responsible for tissue necrosis. It is responsible for anti complementary, means uh, complement system. We will study complement system, classical and alternative pathway of the complement system. So the lipid A or endotoxin has an anti-complementary role. It has anti-tumor action, right? Anti-tumor action. It has some B cell mitogenicity property, means it causes proliferation of uh, B cells, B cell mitogenicity. So it has many pathogenic roles. It leads to pathogenesis of many uh, disease. Okay, another part of the LPS is core polysaccharide. Core polysaccharides. Core polysaccharides is, as the name itself implies, its meaning. Polysaccharide means it is made up of sugars, sugar moieties. It projects from the lipid A. Okay, it projects from lipid A. Third structure of LPS is O antigen. You can call it O antigen, or you can call it somatic antigen. Any other name for it? O antigen, somatic antigen, or we can also call it O side chain, right? O side chain. It induces what? It induces antibody formation. Important. It induces antibody formation. Okay, antibody formation. And it is polysaccharide in nature and varies in composition between the various bacterial strains. It is also responsible for the serotyping of bacteria. Okay, it is also responsible for the serotyping of the bacteria. So we will see the structures. Let me show you. Yeah, this one. Here you see this is positive and this is negative. See, in the positive we studied that there is peptidoglycan layer, then there is your plasma membrane, which is a phospholipid bilayer consisting of the internal proteins and transmembrane proteins, and then we have the teichoic acid, which may be the cell wall teichoic acid or lipoteichoic acid. This is the lipoteichoic acid which traverses across the entire peptidoglycan layer. On the other hand, this is this is lipoteichoic acid, and then there is a teichoic acid which is just buried in the peptidoglycan layer. You can see the thickness of the peptidoglycan layer is quite thick. It is quite thick, but otherwise the cell wall is made up of only teichoic acid and peptidoglycan layer in the gram positive bacteria. Here see, and gram negative bacteria, peptidoglycan layer, where you can see the peptidoglycan layer. Here it is. You see, this is peptidoglycan. Peptidoglycan C. This peptidoglycan layer is hardly, hardly a two-layer thickness. On the other hand, in the gram positive bacteria is many layers thick, right? So that is a major difference in negative and positive. But again, as I said, gram negative bacterial cell wall is quite complicated because it consists of outer membrane. This is your outer membrane, and it consists of the lipopolysaccharide. Lipopolysaccharide is this thing. Okay, lipopolysaccharide has three components: lipid A or endotoxin. These are lipid A. Lipid A is it is endotox. It provides the endotoxic activity. Then it is made up of the core polysaccharides. It is made up of the O specific side chains. O specific side chain is on the outer surface of the outer membrane. Okay, then we have outer membrane proteins. We have outer membrane proteins. Outer membrane proteins are buried in the outer membrane. Okay, so this is the basic cell wall structure of gram positive and negative bacteria. We have already discussed the peptidoglycan layer of positive and negative. This is positive and this is negative, right? So in the positive, there is NAG and NAM unit which are cross-linked to each other with the help of the tetrapeptide, and the tetrapeptide consists of L-alanine, D-glutamine, L-lysine, and D-alanine, right? And there are cross bridges with the help of pentaglycine bridge. While in the gram-negative bacteria, there is nothing like pentaglycine bridge, and the tetrapeptide. This tetrapeptide is also chemically different because the third amino acid is not L-lysine, instead it is mesodiaminopimelic acid. Okay. Okay, so we will move to another section. Next thing is be demonstrated. Cell wall can be demonstrated by various techniques. We will not again go in detail. That is plasmolysis. That can be differential staining. There may be some staining techniques. There may be a specific antibody that uh, we can demonstrate, right? We can demonstrate specific antibody. Then we can use the electron microscope. Okay, that's not important. Another structure is cell membrane or plasma membrane. Cell membrane or plasma membrane, we will go very quick about the cell membrane because this is not important for us from USMLE point of view. So very quick, cell membrane or plasma membrane. It is a fluid. For the structure of the cell membrane or plasma membrane, we have a very famous model called fluid mosaic model, and it says that this plasma membrane or cell membrane is 5 to 10 nanometer thick. Okay, and consist of the phospholipid bilayer proteins, and proteins may be integral proteins, peripheral proteins, and there are no sterols, no cholesterols except the mycoplasma. And function of the cell membrane is like it is a semi-permeable membrane, right? It is a semi-permeable membrane. It is a semi-permeable membrane, and so act as a osmotic barrier. Okay, it is a semi-permeable membrane. Another action is it transports the act as a transport system. A transport system, means it helps in transporting the various various electrolytes and proteins and enzymes. Okay, proteins and enzymes. Then another thing is it is a special receptor molecule. Cell membrane itself is a receptor, and it has metabolic activity itself, metabolic process, right? Metabolic process. So cell membrane. Then comes cytoplasmic matrix. Cytoplasmic matrix, we will not go into detail of the cytoplasmic matrix, otherwise it lacks membrane bound organelles in the bacteria, unlike in the eukaryotic cells where the cytoplasmic matrix consists of the membrane bound organelles. In the bacteria cytoplasmic matrix जो होता है cytoplasmic matrix, it consists of 70% water. It consists of ribosomes, some storage granules called inclusion bodies. It consists of mesosomes. Mesosomes are nothing but the cell membrane invaginations. It consists of various cytoskeletal structures. If we talk about the ribosome per se in bacteria, uh, ribosomes, they are the site for the protein synthesis. The ribosomal RNA plus ribosomal proteins are the combined structure that participate in the protein synthesis. They have a particular Wedberg unit called 70S. 70S is made up of 30S smaller subunit, and the larger subunit is 50S subunit. 30S plus 50S. Then comes the intracytoplasmic is important structure in the plasma membrane. It is a plasma membrane plasma membrane invagination, right? Mesosome is a plasma membrane invagination, and it is more prominent in gram positive bacteria. You can say prominent in gram positive. It is prominent in gram positive. Function, I will say mesosomes acts like, mesosome basically acts like mitochondria. Now it is easy to remember or understand what is the function of mesosomes. Mesosomes, M stands for mitochondria, M stands for mesosome. Easy to remember, as the mitochondria plays an important role in the cell respiratory function in a eukaryotic cell. Mesosomes plays the same role in bacterial cell. It also participates in cell wall formation, and it has some role to play in the chromosome replication. We will not go into detail again. Next structure is nucleoid. In bacteria, there is no true nucleus, no nuclear membrane, no nuclear membrane, no nucleolus. So what is there? Uh, Bacterial nucleoid is irregularly shaped region. It contains genetic material. We call it nucleoid. Okay, we call it nucleoid, and it consists of single haploid chromosomes. Right? It consists of single haploid chromosomes and supercoiled circular. I will say supercoiled circular supercoiled circular double stranded DNA. Okay, and no basic proteins. There are no basic proteins like histone proteins. No histone proteins. No histone. Okay, so structure of the nuclear or genetic material in bacteria is pretty basic. It is nucleoid. There is no nucleus, no nucleolus, no nuclear membrane, no basic proteins in the associated with the DNA. DNA itself is highly coiled structure, super coiled structure, circular DNA. It is circular DNA, right? It is circular DNA, double stranded DNA, and except the Vibrio cholera. You keep it in mind. Vibrio cholera is a bacteria where there are two chromosomes. Two chromosomes. Otherwise, in all bacteria, chromosome is haploid and single chromosome, except, so I will make a star mark, remember it, it is an exception. Vibrio cholera has two chromosomes. Okay, there is one more important structure, we will come across it in when we will go through the bacterial genetics, that is plasmid. This is this is plasmid. What is plasmid? Plasmid is extra chromosomal DNA. Extra chromosomal DNA. Extra chromosomal DNA. Okay, today's topic here, and we'll cover the remaining portion of the topic, same topic, which will include bacterial growth and very important part of this topic, virulence factor among the bacteria, in our next video. So guys, if you like this video, please subscribe, like and comment to my YouTube channel. Thank you.